formula which we use to look for linkage. If a family comes along, let us say that you identified a disorder or something, a monogenic disorder. This linkage analysis won't work that easily with multifactorial. If it were to be a single gene disorder, such as cystic fibrosis or any other disorders that you are interested in, what you can do is you can go to the family members and collect all the blood samples for the entire family, from siblings, parents, and possibly grandparents, and if they have siblings, they have children, they have children. So if you can collect all the family members, then what you're going to do is toss a coin. It is like odds ratio. Like, for example, if you go to a gambler and say, I have got a 3-to-1 odds to win. What does that mean? That means whatever $1 you put in, $3 will come to you or something like that. Is that correct? So that is the odds. So here, you are going to bet on whether the individual is likely to have linkage or no linkage. That is the kind of the analysis that you do, but that is based on the number of recombinants that are present in the population or in a family. That is, there is an equation here. This is the likelihood ratios, the likelihood of the data if loci are linked at a particular recombination fraction, theta, and the likelihood of the data if the loci are not linked. We told that the best linkage of theta should be equal to 0. If theta is 0, which never happens, essentially the two base pairs are linked. That means if the recombination distance is only one base pair, that means the theta is going to be absolutely 0. If the disease locus is 12,070 base pair, 12,071 is the polymorphism, they are linked, tightly linked, right? Just very, so at that stage you will have a theta will be definitely 0. But if theta is 5, that we showed in the previous slide, the curve, that means there is no recombination at all. That is theta 5. So that is the likelihood of the data if loci are unlinked is theta half. So now what you can do is you can go back and look at another equation by taking the recombinants and non-recombinants from this family. Okay? With respect to this disease locus, this is all absolutely, there is no recombination here, but there is a recombination here, right, in this area. So if you take those lists and then substitute the number of recombinants, let us say you get 6. How do you count the recombination? Let us say you run a polymorphic markers. Okay. In family 1, let us say you had 2 bands. And in another individual, you had another band, something like that. To begin with. But then if the recombination occurs, this band from family 1, like a maternal band in one individual when it is inherited, this band may appear in the population and so on. So that is called recombination. So by looking at the pattern of the DNA bands that are inherited, like polymorphisms, you could tell whether it is polymorphic, they are polymorphic bands or recombination occurred or not in the meiosis. Okay? Just like what you saw in the previous slide, you know that if you run a markers for these loci, you are going to tell that there is a recombination of these markers, whereas there is absolutely no recombination of these markers with disease. Okay? That is, you follow a person who has a disease and who follow heterozygotes and things of that kind, and you can get those recombinations from them. So that is called 6 of the recombination here. Now what you do is you have the same formula. There is a likelihood of the data if the loci are linked, or 1 theta to the power of 0 times 1 minus theta to the power of 6. Okay? But now why did they convert into the log? They made a logarithm of, logarithmic one, this particular. That is because if you just compute only, if you are in the log scale, then you can add the data from multiple families and just add up the fraction. That's all it is. Okay? You can also do the regular way and then add up, but the addition will become a little bit more complex. You have to go to the tables and look at it. Whereas the logarithmic one is pretty easy. Therefore, the theta 0 times 1 is theta minus 3 to the power of 6. But now let us substitute. Now this is called parametric. At this point, you have to use likelihood of the data at various fractions of theta. That is, assume that theta equal to 0. Assume that theta equals to 0.001. Assume theta equal to 0.002 and keep on increasing. And now you are going to calculate, based on the recombinants, you are going to calculate the recombination, the likelihood of the z values for every fraction of theta theoretically. Okay? That's what you're going to do. So take this equation and put theta 0.1, 2, 3 and so on and so forth. At that time what you're going to get is a fraction of at 0 recombination and 0.1 and 0.5 and so on, you are going to get a fraction of 1.8 and all these things are there. So at this point when you say, hey look, at that fraction you got 1.8, but for example if you got in the value, this is the value that you got, that means the recombination fraction would be 0.10. Okay? So now look at here. In anything that is above 3 is considered to be significant linkage. Above 3 means you are going to get somewhere close to a little bit higher value, like somewhere here. Okay? So now in the family 1, you can calculate like that. In family 2, you can calculate because it is a logarithmic base. You can simply add up this and you got a value. So at 0.1 fraction, you have almost like 3. Okay, that means it is greater than 3. 0.1 means what? 0.1 means, what is that? 0.1 is recombination fraction. I told you if it is 0, essentially it's tight linkage, right? But then 1 centimorgan is equal to 0.1 fraction, okay, tenth of that. So if you find that 0.1, that means you are in 1 centimorgan away from the disease locus. So that means if you get a large score of a 3 at 0.1, that means you have relatively significant linkage. Okay? So now if you know that the locus is whatever this marker, that is polymorphic marker, is 1 centimorgan away from the disease locus. That's what this large score is telling. It's also called logarithm of odds. It is called large score. If large score is greater than 3, it is an evidence for linkage. And once you find an evidence for linkage, what do? Now you got a disease locus. The disease locus is D, and you got a marker locus which is somewhere here, and you know that the distance is approximately 1 centimorgan. Okay? But it could be disease locus here, and the marker can be here. So both the possibilities. You can still have 1 centimorgan distance. It doesn't tell you which side it is. Orientation it doesn't tell. What all it tells the recombination fraction is that the distance from the disease locus is smaller between the polymorphic marker that you have used. So now what you have to do is you obviously have to go from marker locus, you know, because you have a marker, therefore you have the gene, not gene, you have a DNA fragment for that location, same location here. So you have to go from here to there by genome walking. That is, you take a fragment, you just do the library, you get this clone. If you have lambda genomic libraries, we talked about all the libraries in the first, second class or third class, or if you have a back clone, you clone 200 kilobases of the gene. So now, you got 200 kb, okay, and how will you go? You have to walk this way. Suppose if you walk this way, yes, you are going to get the disease gene in 1 million base pairs. But you have got 2 ends for the back clone. So you can use this N probe, okay, and then again use the library. You can clone one more, so you'll have overlapping clone. Another overlapping clone, another overlapping clone, like that. So you can keep on walking. Because it's 200 kilobases, how many walks you have to do? 1,000 kilobases is 1 million base pairs, correct? I mean, so 1 kilobase means 5 walks. 1,000 means a lot of walks you have to do. It is tremendous distance, not a joke. Furthermore, suppose instead of walking this way, you walk it that way, you won't find the disease gene. Okay? So how will you deal with it? So the way to deal with that is go back to the same family and use additional set of polymorphic markers. That is, you use another marker which is on this side of this. Therefore, you have to again randomly look at several markers one more time and find a marker which is linked to some other distance, like maybe 3 centimorgans, okay? Let us say 3 centimorgans. So that 3 centimorgan probe may have another gene which is located somewhere here. So that tells you some knowledge of the gene or some other fragment will be there. With that knowledge, you can tell that this is in this direction, and then you know how to walk there. So that is one way to circumvent the problem. But if you don't have any information at all, tough luck. You have to do this kind of a brute force walking. There is no choice. Okay? That means you have to walk 1 million base pairs, find a gene there. That gene won't be disease gene. Walk the other way, find a gene there, and then see whether that gene correlates with your disease. That is the way it was done in the olden days. Francis Collins, who was a colleague of mine, was actually a junior postdoc at that time when I was at Yale, developed a technique where instead of using these probes, you have to take the back and then clone them, end clones. What he did was he circularized this fragment of back, and therefore these fragments are going to be attached to each other, right, when you ligate it. So you can get both fragments at one time. Therefore, he called it chromosomal jumping. Instead of walking, he called it chromosomal jumping. And in fact, he went ahead and used the same technique at that time to clone the cystic fibrosis gene. He was the one who cloned first time cystic fibrosis gene when he was at the University of Michigan. Now he is the director for National Institutes of Health at Washington. Okay? So that is the way the genome walking and jumping were done. But now, and of course various other genes have been, at least now several tens of genes have been located by using this linkage analysis. So this is exactly what we are talking about. So you have a family, because of the way that it is migrating, you can tell whether recombinants or not recombinants based on the polymorphic marker. But in this example, what he is telling you is how the phase knowledge of the phase, we talked about the phase, one repulsion and coupling, right? The knowledge of the phase is going to help. In this family, you do not have information about these individuals here. So these are affected individuals, but you have the knowledge of how the disease gene and the marker gene are associated here. Okay, capital D and capital M, D and M. But unfortunately, because there is no knowledge here, but in this family, look at here, there is information here also. So that tells you that the D and M are in cis or in coupling. Whereas here, you cannot tell that. Although you wrote it there, that doesn't mean that is in coupling. It could be the other way around. Okay? It could be the other way around. Therefore, in this individual, you have to tell that it could be either non-recombinant or recombinant. We don't know, because we have no clue about the phase information. Whereas here, in this case, because the information is known, that is traveling together, and therefore they are all non-recombinant. There is no recombination between D and M. That's what you can decide. Whereas here, it could happen both ways. You can tell that it could be recombination, or there is no recombination between the disease and marker locus. So if you have a phase information is lacking, then what you do is you do the same formula again and compute the same large scores. But unfortunately, because the phase is unknown, you are going to get the large scores differently compared to the family. Same family, if the phase is known, if you substitute, you're going to have a differences between the large scores. See, knowledge of one single individual with a simple phase information, because that is traveling together, has added, see, after all 5 recombinants before, then you add one more family member, and one additional recombination is going to dramatically increase the large score. That means, if you have a family of 10, all of a sudden you find another family of one individual information also is useful for recombination fraction. Therefore, any time when you get a new member, knowledge of the new member, you go ahead and add it, and thus you can correct the recombination fractions in this game. So now you assume the same formula here, but he has simplified it. This particular formula is actually theta to the power of 0 times half of 1 minus, because this is going to be half and half in a phase unknown situation. So he modified the equation and arrived at that particular figure, okay? So it is for, because there are 3 non-recombinants and 3 recombinants, out of 6, 3 are non-recombinants and 3 are recombinants, that's what you are assuming. And therefore, instead of previously 6, you are using the figure 3, and using that you can calculate for the phase unknown. But the moment you get a phase known, then you can do a different job. Okay? But in case of pedigrees like hemophilia, you don't have that problem, because these are X-linked disorders. And furthermore, you also know, except for one region in the X chromosome, which has got high recombination between X and Y. What is that area? We talked about there is one region, one gene located in Y chromosome that can easily recombine, and that will confuse you for certain disorders like 46X and things of that kind, 45X. What is that disorder? What is that? That is correct. SRY is the only region which we know that it is highly recombinogenic. Okay? Rest of the areas on X chromosomes do not exchange with Y. That means whatever gene that is present in that male, if it is what to be X-linked, that gene will not undergo recombination. That is, any marker locus will not undergo recombination. Therefore, obviously the phase of that individual is always known. You don't have to worry about the phase problems. So you can very easily tell that HM, even if you don't have that male, that this particular HM is going to be definitely linked, okay? So that is the way you can easily compute for this HM, and you know that there is linked, and therefore it's easy to map X-linked genes. In fact, a postdoc of mine, I told you, Warren, what's his name, Warren, this fellow cloned the Fragile X gene. That is because it's easier. I cloned factor 9 in those days, and Fragile X was very close to factor 9. So you usually probe and walk to Fragile X locus, okay? So that is what happened in the X-linked chromosomes. So this is the model base. That is, in other words, you use these equations and model it, because the recombinants present in a family are going to be limited number. So you assume theta equal to 0 until you get that values large score of 3. That is why it's called parametric. These are parametric because they are all based on parameters, and that is why they're parametric. And a non-parametric linkage analysis is, you could use the SIB pairs, concordant SIB pairs as well as a discordant SIB pair method. See, this is all useful for qualitative traits. So what you can do is the polymorphic markers shared by the SIBs you can more frequently are noted, and therefore it suggests the linkage. So you don't have to worry about family-based studies. These are non-parametric. You're not really calculating any large scores, but what you are going to look for is those markers which are shared that suggest the linkage. Whereas the SIB pairs which are discordant, let us say this is all for qualitative traits. It's very easy because you don't have to worry about quantitative methods. It's a qualitative trait, and you can follow it by the SIB pairs which have got some kind of polymorphic markers which are shared. Whereas if it is a discordant SIB pair method, that means if there is some kind of a quantitative trait, like you've got a Gaussian distribution, you can take 2 standard deviations away from this area. So that means, let us say, we talked about the diabetes, right? If it is a glucose level 120 plus or minus 10, if it is a 2 standard deviations, you are talking about 110 plus 2 standard deviations would be 20. So that which is beyond is the one which you are going to look for, okay? That which is common there. But in this case, what happens, anything that is considered discordant is having a quantitative trait which is 2 standard deviations away from that, okay? So you could use that. So that also you can show that if you find that kind of a value and associated with the markers, that is called discordance for quantitative traits, that also shows an evidence for the linkage. So these are some other methods where you do not have the ability to collect the family members, and therefore you could use this kind of a method by comparison of the SIB pairs and say, hey, look, this probably is linked, but there is still not an evidence for the linkage. That is causal linkage to the disease locus. That's all it can tell. then, therefore, they called it, instead of calling it, although it's called non-parametric linkage studies, it is truly an association study. Okay? In association studies, there are furthermore other association studies out there. It's called case control and a cohort study. Okay? That is a group of a population, a group of people you can collect and look at what markers are linked to the locus. So in this case, what they can do is, if there are two alleles in a population, okay, and you have people who are suffering from a disease with a particular allele, that is, with one allele, these are the number of patients. Let us say there's about 900 patients you collected. They all have one particular allele. But in the same, another patient population, you can take the same population and say without the allele is the total number. And in the controls, you can go to the control people, another collection of 1,000, and see with allele what happens to that number, and then you have got equations where these numbers can be computed, and you will get what is it called, an information about the disease odds ratio. So that is the disease odds ratio. Okay. So this is also another ratio called relative risk ratio. So you have that particular equation. You can look into that in the book. So let us take a practical example. We talked about the prothrombin mutation before, right? The prothrombin mutation is that 20/210G is converted to A. So that is what prothrombin mutation. That is one allele. If that allele is present in individuals who have got a cerebral venous thrombosis, CVT, about 23 of them. That allele is absent in 97 patients. Whereas this is 4 and 116. Therefore, it is really, whatever this allele is present and that is absent in these many individuals, therefore it will tell you relative percentage of individuals who are at risk for the presence of that allele. That is called relative risk ratio. So if you calculate the odds ratio, you are going to get approximately 6.9. That means, it's almost like 7%. Okay? So that is what the odds ratio is telling you. By using that odds ratio, okay, if the odds ratio is greater, that means you can tell that that particular polymorphism is linked to the disease. Okay? So we talked about an example which is already known, that's why you know the ratio, but otherwise you can take any allele, doesn't matter whether it is prothrombin allele or some other allele like cystic fibrosis allele. As long as you calculate those frequencies, you can tell that it is there possible. So we basically touched upon genome-wide associations in the last class a little bit. And there are several regions, several genes have been cloned by using this positional cloning. But what are the genome-wide associations? Genome-wide associations are, let us say that you have, 1 million SNPs, right? Out of 3 billion base pairs, you've got 1 million single nucleotide polymorphisms which are present between 6 different populations of individuals, African Americans to Japanese. They've already computed that, and you have what is this called HapMap. HapMap has got certain areas where they are all recombination hotspots, and that where the recombination doesn't occur, they have already mapped. That means those polymorphisms are already tightly linked. But out of these million polymorphisms, you cannot really do all million analysis. It's impossible. So they have what is it called tagged polymorphisms. What they have done is, see, we talked about HapMap. In the HapMap, we have one particular group here we showed in the slide, right? So that is the cluster 1. That is, and there is one more cluster here that is a cluster 2. But among this cluster also, you could select, I showed you 14 nucleotides, right? 11 nucleotides on one side and 4 other nucleotides on the other side. You could select out of the 11 a very common one, just not all the 11. You cannot study all 11. So you select one of the common ones here, and you select one other common ones here, which are approximately situated 1 centimorgan apart. Okay? If you want a million nucleotides, there are too many numbers. So instead of doing it as not 1 centimorgan apart will be too much, let us say you select markers which are about 10 centimorgans apart or 1 centimorgan apart. That way you could use that already existing information to go back and look for single gene problems. There's no big deal because now you don't have to worry about cloning, chromosomal walking, jumping, etc. because all the genes are already there located, so you can go back and look at it very quickly. That is one way. That is the linkage standard analysis, but with the help of the human genome. But on the other hand, if it is a multifactorial inheritance, like age-related macular degeneration or Hirschsprung disease or whatever, there are lots of disorders which we know about polygenic. For these disorders, how will you do? The best way to do it is go back to the same method of collecting those who are absolutely, you are sure that they are homozygous for the disease, and you are sure that they are coming from one homogeneous population, and collect several individuals and pool their cell DNA first. The very first thing that you have to do is to go back and collect a lot of DNA samples from already individuals. If you don't do that properly, you will have a mess at the end. So selection of patients is extremely critical for this kind of a genome-wide analysis. So after collecting the DNA from these individuals, pool them, that means you have now multiple individuals who are having possibly various types of polymorphisms here. Okay? These polymorphisms, they have already designed specific oligonucleotides to these polymorphisms. That means if there is a G, this will be a G containing oligonucleotide. If there is some kind of an A polymorphism, that will be a containing oligonucleotide. Now, all these oligonucleotides for these polymorphisms have been already put on a microarray chip. So that is oligonucleotide 1, 2, 3, 4, like that. There are million oligonucleotides you can put on one slide. It's no big deal. Now, you go to the genomic DNA that you isolated from these patients. Whatever way you want, you can do PCRs in those areas. So this area, you can do PCRs here. So that DNA will be amplified. That means you can amplify thousands and thousands of fragments of DNA from these patient DNA that you have already collected. And now you probe them directly onto this slide. If the patient DNA matches perfectly with the G, it is going to hybridize. Okay? There are various methods where you can play tricks of hybridization. What? By temperatures you can vary, or by extensions you can vary by DNA polymerizations. Whatever it is, I'm not going into details of the methodology, but the simple idea is, if it matches perfectly, it is going to light up. If it doesn't match perfectly, no hybridization signal, or whatever the signal that is coming out. So by looking at it, you can tell that spot number 1, 10, 20, 30 are all present in the individual, whereas other spots are not. Therefore, several markers at the same time can be identified that they are all causally associated with the disease. So it doesn't matter how many loci. Let us say the polygenic disorder like Hirschsprung disease may have 20 different loci. We don't care. But those 20 different loci, if they are closely linked, or at least associated with the disorder, you are going to get haplotypes which are associated with this particular disorder. So you're not going to get one marker, you're going to get multiple markers. Okay? Using this kind of a methodology, they have already collected in the past. It's actually the concept started in 2001, but within an 11-year period, believe it or not, in the entire world, they have collected almost like 1,400 different polymorphic loci, and almost like 700 disorders have been already attempted. But unfortunately, still, they have not identified the genes which are causing the disease. They got associations. But there is a great study at Duke done recently, not in Duke, the University of North Carolina. In fact, I heard her talk only very recently in a conference. And what she did was, she took the type 2 diabetes as an example to see whether she can identify. So what happens in this kind of a game is that you will get a lot of markers. So they just plot like this, chromosome 1, 2, 3, 4, and so on. So this is the way the plot will look like. I don't think that is present in your textbook. you're going to get all these dot plots. These are all positive markers, but some markers will